All right. It's now time to enter our first theme with health from life. We'll be looking at many areas. Previously, in the past, we looked at chemistry, and I know that Martin Koplos, our Nobel Prize friend, is in the audience today, so welcome, Martin. Very happy that you are here. And today, we're going to look at two other areas. Biology first, status on one of the leading causes of death worldwide, which is cancer, accounting for 1 in 6 deaths today, and could impact almost 30 million people a year by 2040. And then medicine, with the rise of AI-enabled technologies coupled with a virtual world. To start with, biology first. I am particularly delighted to welcome our first guest. And it's, in my personal career, also a moment. Professor Weinberg is one of the world's most, you won't like it, but I'll say it anyways, most prominent cancer biologists and researchers. He has changed the conceptual paradigms around cancer, and I must say, inspired a whole generation of scientists with his seminal works on whole mass of cancer, and you will see up to even what we did at the very beginning in Dassault Systèmes when we started the life science and healthcare journey. In fact, what we did, thanks to him, was, in systems biology, the ever first virtual twin of a cell, based on his research. Please join me in welcoming Dr. Robert Weinberg. And you have the clicker. Thank you so much.
Thank you, Dr. Johnson. I was pleased and flattered to be invited here to speak after Dr. Chalez, Dr. Johnson. I'm here as a representative of the vast community of people who do cancer research, or as some cynics say, more people are living off of cancer than dying from it. It's not really true. And I'm here to give a realistic, not an optimistic view, but a realistic view of the goal of reducing mortality from cancer. Where actually are we in terms of objective facts? In 1971, then President Richard Nixon started the war on cancer in this country. And of course, the critical question is, where are we now, 52 years later? How much progress have we made? And how many impediments still stand in our way in terms of being able to conquer this disease? And saying that, I make reference to the slide here as the beginning of my discussion to show you the age of onset of most human cancers. And what you can see from these two graphs is the fact that cancer is largely a disease of old people. One exception to that is breast cancer, which starts early. But this has important consequences of how we think about this disease. If we look in this country, at least, at the number of people who are over the age of 65, you can see that, as is the case in all Western countries, the number of these aged people, I among them, is increasing progressively. And what that means is that deaths from this disease of living older actually can go up and will go up continuously. The number of deaths from cancer will go up continuously for the next decades, independent pretty much of how we organize our response to it, our therapeutic response to it. One question one might have is, how much success has there been in reducing mortality from cancer over the last 50 years? And here you can see some of the trends in cancer, in this case between 1930 and 2006. And what you will notice is that many of the cancers, the death rate, age adjusted, is pretty constant. It's gone down a little bit. The major success story is death from lung cancer. And many cancer organizations advertise this significant reduction in deaths. But as you will imagine, the credit for this doesn't go to improved therapy. Instead, the credit goes to the fact that more and more people are stopping smoking. And this leads me to an obvious point, which is that if we're going to reduce the incidence or the mortality from cancer, if we really wish to do so, then changes in lifestyle will be vastly more important than anything I or my colleagues can do. Here, this kind of reduction in mortality that you see associated with breast cancer is extraordinary. And if my colleagues and I could achieve anything like that in terms of reduction in mortality, we would be ecstatic. In other words, prevention is ultimately going to be far more important than treating the disease in terms of reducing mortality. Cancer researchers don't like to hear that because prevention is ultimately a psychological field rather than being a scientific research field. How have we arrived at where we are right now? Here is a very important and seminal series of observations made by a man named Bruce Ames, then and now at the University of California, Berkeley. And what he advertised in 1975 was that if he looked at a whole series of different carcinogenic compounds, well, I was very grateful for the laser pointer. Whatever. At MIT, we have these problems too. As you can see from the graph, as he discovered then, compounds, carcinogenic compounds, have greatly differing degrees of DNA damaging capability. We call that mutagenicity. And their ability to damage DNA is strongly correlated with their damage to induce cancer. And that led to the obvious notion that damaged DNA in cells throughout the body is responsible for cancer. It creates the genetic paradigm. And that if one can understand the DNA damage within individual cells, one has made a major step forward in trying to understand the process of cancer. The fact is that this genetic paradigm, as I will refer to it repeatedly, said that cancer cells are mutant, that is, they carry mutant genes, damaged genes. And secondly, and equally importantly, these mutant genes are actually causally responsible for the aberrant behavior of the cancer cell. And here is work that I participated in 1982. And what you can see here is the demonstration in the DNA of a human bladder carcinoma that a single change in the base sequence of the DNA, remember that there are 3 billion bases of DNA in the human genome, a single base pair change, and you can imagine that my laser pointer works, which it doesn't, but you can see in the top right that this single change in DNA created a profound change in the biology of cells, and that mutation was on its own causally important for the aberrant behavior of cancer. And observations like this led to this genetic paradigm. If we understand the genes within cancer cells, then in fact we can understand how they behave aberrantly. Is there anyone in the audience who has a laser pointer? I feel like a stewardess on an airplane. Are there any physicians here on the plane? Anyhow, en tout cas, since that time, the genetic paradigm has exploded since the early 1980s. And what we see now is that there are now 291 genes in the genome of human cells that, when damaged, can actually help to create cancer. So the notion that this is going to be simple is already complicated by the fact that these genes act in various combinations. And if you understand the mathematics of combinatorial interactions, Miss Johnson, you get much credit for this. She's not related to Patrick, by the way. Thank you for that. She gets more credit than I in making this coherent. So here we have a large number of genes that act in various combinations, and this is already analytically almost impossible to understand how these genes act in combinations to create mutant cells that create cancer. In fact, 1% of the genes in the human genome, when mutated, contribute to the formation of cancer cells. And individual cancer cells have multiple mutant genes that conspire with one another within a given cell to actually create cancer. Over the last 40 years, my friends and I have developed this wiring diagram of the cell. This diagram shows a whole series of proteins. Their names are in black and red here. And they are responsible for signal transduction, for the processing of signals that go into cells, and for informing a cell as to whether it should grow or not grow. This is an extraordinarily complex wiring diagram, but it only represents maybe 10 or even 5% of the total complexity of the intracellular circuitry that is perturbed by these mutations. Here, for example, is the gene and protein that I described before. I will tell you, as a simple fact, nobody understands how this circuitry works. In other words, it is complex beyond measure, and we do not have the analytical power to figure it out. Given the combinatorial actions of all of these signal transducing proteins that require an understanding of amplification, of negative feedback, and so forth. The physicists and the mathematicians and the proponents of AI say, just show us this wiring diagram and we'll figure it out for you. We will show you how this works, how it actually is supposed to work. And as I often say when I talk about them, I will often say, if you shake hands with such a person, you should count your fingers afterwards. They are not to be trusted, because this biological complexity, which has been developed by accretion over the last literally 2.5 billion years, is complex beyond measure, at least as we currently understand it. Here is one such example of the genetic paradigm. This is an example of a protein which is aberrant in chronic myelogenous leukemia, whose mutated gene that directs its structure was discovered already 3 decades ago, and a successful interdiction of the signaling by that protein. This protein here in green drives the proliferation of the cells of chronic myelogenous leukemia. Through extraordinary organic synthesis, one was able to develop, Ciba-Geigy and Novartis and other companies, a drug called Gleevec. And Gleevec, as you can see in the bottom of the slide, has an extraordinary effect on patients who start out in the early phases of chronic myelogenous leukemia, which in other situations would surely have killed them within a year or 2 or several years. This is an extraordinary success story. But for better or worse, it represents the poster child of success, because everybody seeing this kind of success said, we'll make more such inhibitors and we'll interdict the function of yet other such proteins. Look at this, how this compound, Gleevec, fits into a pocket within this aberrant signaling protein and shuts it down. So this represents a major success story. And following up this, there was a whole campaign over the ensuing 10 to 15 years of developing other similar agents which could also shut down aberrant signaling proteins, with the notion, with the hope, that these too would be as curative as Gleevec was. And here we will see that there's a whole series of other such compounds. You see them listed here to the right and to the left, which are able to shut down these signaling channels, doing so very successfully and with great specificity. At the same time, there have been successes in using the immune system, in mobilizing the immune system. In this case, a major success in the development of the monoclonal antibody Herceptin, which can prove to be very useful indeed in essentially curing many women who have HER2-positive breast cancer. So this is not a minor step forward, but there's a major fly in the ointment. And that's illustrated by what you see here. Because when you start in the development, the genesis of a tumor, the tumor is pretty homogeneous, but as years grow on and the cells divide and multiply, as this proceeds, the cells within a given tumor become increasingly heterogeneous. They become increasingly diverse. And one cell type and one genome cannot represent the biology of the entire tumor. There are subpopulations, as you can see over here, which are quite diverse. And the consequences of this are that when you look at a tumor will look more like this. That is, it's not a homogeneous population of cells. And if you treat this tumor with many of the drugs I just showed, then what you see is that many cells in the tumor will respond, but there will be outliers here or here that are no longer responsive to the tumor. This genetic diversification, in fact, represents a fatal blow to those who would say, if we just develop this drug, we will cure human cancer, because cancer cells and tumors become more and more different from one another as tumor progression proceeds. Here's one exact representation of that. Here's Gleevec again, fitting into the pocket of a protein I showed you before. Here's the drug. And what happens in some patients is that the patients, the cancer cells in those patients develop a mutation which creates a bump here in this pocket, and this pocket no longer accommodates the drug, and as a consequence, those cancer cells are no longer responsive to the drug. And just as an example of the fact that cancer cells can evolve defenses against various kinds of agents that are launched against them. And as a consequence, these brilliantly conceived drugs, I showed you a list of them before, in fact can extend the lifespan of a cancer patient by 6 months, 12 months, sometimes several years. But sooner or later, the cancer comes back. And here you can see the reason why. And this is a problem we have not yet succeeded in overcoming. I'm not trying to be optimistic or pessimistic. I'm trying to be, in this case, realistic. What are the problems we're confronting? So developed resistance is clearly one such way by which cancer cells will outfox us. But what are the other problems? Here's the second problem about the genetic paradigm I told you about before. If you look at a tumor under the microscope, what you see is that the tumor is actually a complex of tissues. Here are cancer cells in the lower left of this particular micrograph, and there's a whole series of other cells that are recruited to the tumor mass during the course of tumor formation. These cells are called collectively the stroma, and they too influence profoundly the biology of the cancer. And their influence cannot be understood by looking at the DNA of the cancer cell, that which is only in these cells over here. So that's another major complication. The next one I've just told you about, the circuitry. We really don't understand how the circuitry works in aggregate of a single human cell. Keep in mind we have 3 times 10 to the 13 cells in the body, and each one contains this complex circuitry. And I don't see any change in the immediate future in understanding and being able to predict how a cell is going to behave on the basis of our understanding how this circuitry processes signals. It's extraordinarily frustrating, and it's not one that's guaranteed to be solved in the immediate future, even by the much touted AI. And even if you have or have not shaken hands with a proponent of AI. Here's another difficulty we confront in trying to conjure up treatments of cancer. We would like to be able to predict from work in the laboratory, often with laboratory mice, how will actual cancer patients respond to this or that drug in development? Can we predict its efficacy in vivo? We in mice diverged maybe 60 million years ago from one another. We have very similar genomes, but biologically, we're actually very different. It takes 5 genes to transform a normal human cell into a cancer cell, but only 2 in the case of mice. And the fact of the matter is that our ways of predicting clinical response on the basis of preclinical research of the sort that I'm showing here really leaves one often very frustrated, because these predictions are actually often very far off the mark. This is a major problem, as you can imagine. Here's yet another problem, and that is looking at a whole series of patients. Here we've lined up 50 or 60 patients, right to left. Each one is a bar, and their response, their respective responses of the individual patients to the drug, that is to say, the responses of their tumors, actually are, these tumors continue to grow here. This one is stopped dead in its tracks. And these tumors here in these patients, another 20 patients, they actually shrank. Well, that's mildly interesting, but it represents a major problem, because individual patients within a group that are categorized together as one homogeneous group actually behave very heterogeneously in terms of clinical behavior. And that too is a response, is a difficulty. Here's yet another problem with the genetic paradigm as I've announced it. Here, we look under the micrograph at this mammary carcinoma. And what you see here are 3 colors of cells. The cells on the outside are the host cells, that is to say the stromal cells. They have blue nuclei. To the upper left, you see the red cells. Those are bona fide carcinoma cells that reside in one state. It's called epithelial. And the green cells on the outside of the island, in the upper left, are also carcinoma cells, but they've shifted their state from one phenotype, from one behavioral class, to another. And this change, this shift in cell phenotype has profound effects on the behavior of the carcinoma cells, causing them to be very aggressive. Importantly, critically, the red cells and the green cells have identical genomes. What has happened to them is that the green cells have shut down certain genes and turned on others. And again, we can't predict that from knowing the DNA sequence of the cancer cells, because these shifts from the E to the M, the epithelial to the mesenchymal, as is often called in biology, these shifts do not involve changes in DNA sequence. They are not mutations. And yet, it's really extraordinarily important in trying to predict the aggressiveness of common carcinomas, for example. So, in a sense, much of development of curative treatments of human cancer is stuck in the mud at present. You won't hear that from some of my colleagues who would tell you that miraculous cures are just around the corner, but the reality is frustratingly quite different. We have one hope, and this hope may well succeed in developing really widely acting cures for the disease, and it comes from checkpoint immunotherapy. That is to say, the ability of a cancer cell to be attacked. Normally, a lymphocyte attacks the cancer cell. You see this here on the left. Here's the lymphocyte. Here's the cancer cell. The lymphocyte sends out signals that kill the cancer cell because the lymphocyte has recognized the cancer cell and has the wherewithal, has the weaponry to kill the cancer cell. What happens, however, in reality is that the cancer cell will sometimes turn on a signaling protein here, which shuts down the lymphocyte. In other words, it has a defensive maneuver, which is a negative feedback loop, which prevents the lymphocyte from launching a successful attack. Over the last years, one has discovered that one can shut down this negative feedback seen here by adding monoclonal antibodies. You see them here, these monoclonal antibodies. And these monoclonal antibodies break down the negative feedback loop and liberate the lymphocyte from these inhibitory influences, giving the lymphocyte a free rein, as you can see here on the right, to attack the lymphocyte. And this has led to some dramatic cures in the case of melanoma, even lung carcinoma. But the great majority of human cancers are not yet responsive. It is my prediction that over the next years, this checkpoint immunotherapy is going to be greatly expanded in terms of its successes in giving either long-term survival or actual curative responses in common human tumors. But that's a prediction, it's a prophecy, it's an optimism. But to my mind, this is the great hope at present for the future. And if we don't succeed in getting this to work, which I think we will, we're going to be in, as we say in the States, deep doo-doo, in bad shape. So thank you very much for having me. Merci bien pour tout.
Thank you, Dr. Weinberg. Come with me at the center of the stage because I won't let you go. I think we can have a couple of Q&A session, if you mind. I'd like to start, and after that, prepare your questions in the audience. I cannot resist. You've said everything about AI. How do you see the next generation of biologists working between the bench, experimentation, but still simulations, data understanding, and modeling?
The problem is, and I've seen graduate students for the last 40 or even 50 years, they used to do wet biology, they used to do biochemistry, they used to study how these signaling proteins work. And over the last 2 decades, increasingly, the young people have not been so interested in getting their fingers wet with test tubes. They've been much more interested in doing information technology, in studying computer science. Somehow it's intellectually more satisfying. It's not wet, messy like wet biology is. And that is a critical problem because a lot of problems in understanding how the circuitry works, which I have argued is critical to really developing novel kinds of therapies, understanding how that circuitry works will involve doing wet biology, will involve the wet test tube experiments, the messy experiments that we critically need now in order to move ahead. And so I don't know how this is going to develop because the young people, they don't want to do this medieval work on protein biochemistry in order to understand things. They just want to do the bioinformatics, which is going to solve everything. When I was a student, and this was just developing this field. The physicists came in, they said, this is all very interesting. Actually, we physicists are much smarter than you biologists. We're going to move in and solve all these problems. And that was 40 or 50 years ago. Didn't happen. En tout cas.
So experience is always needed.
Experimentation is needed. Experimentation. And experimentation just is messy and it's complicated and it's challenging, but it's what's going to be critically important for moving ahead. And AI, to my mind, is on its own not going to solve many of the problems I've just described to you. There are too many independent variables that even AI will have difficulty in assimilating and processing.
Thank you. Before giving the mic to the audience, I have a second one. You mentioned immunotherapy.
Yes.
Indeed, it provides a lot of hope in the community on many cancers. What about mRNA as a segue to our next speaker?
Well, we're going to hear from Dr. Laughlin soon. I look forward to that. She's a member of Moderna, which has had brilliant successes, as we all know, in creating COVID vaccines. Truly extraordinary success in an area which I was very skeptical of. How could RNA molecules ever actually get into cells and do anything? And I was wrong, thankfully. And she's going to expand on the brilliant successes that Moderna has had in actually using our mRNA technology to immunize people, for example, against different kinds of cancer cells, I imagine. What was the question?
What do you think about mRNA as a hope for cancer?
Well, the question is, can you vaccinate a person? Can you engender immunity against a person's tumor by analyzing the aberrant genes and proteins in that person's cancer cells and mobilizing the immune system using this mRNA technology to specifically recognize those aberrant cancer cells and to attack them and to eliminate them? I think this is very exciting at present. And for me, the whole mRNA experience is very humbling because it illustrates how wrong-headed I was in being skeptical of it. Whether it will be the answer to cancer that people have sought for so long, we will hear from Dr. Laughlin.
Or a stepping stone to it.
Or a stepping stone to it. We have to keep fighting. We cannot be discouraged by some of the complications I told you about, but we need to realize that they exist. And simply turn one's head away from the complexity, the complex biology of real human organisms, I think ultimately is fatal in terms of being able to really deal successfully with the disease at the therapeutic level.
Thank you very much. Any questions from the audience at this stage? Don't hesitate, don't be shy.
Thanks a lot, Professor Weinberg. Your work is very special to me. I did my qualifying exam for my PhD 10 years ago, studying the 6 hallmarks of cancer that you have issued. So my question for you is, based on the understanding of cancer now, how should clinical trials evolve? As of today, they would still be on a given organ, in a given line of treatment, and with what you told us about the complex circuitry, but also immunotherapies, should clinical trials shift to new methodology, new protocols?
Well, I'm not expert in that area, but my instinct is that there are ways of getting information from clinical trials that are much more rapid, more economical than we're currently pursuing. Clinical trials are horrendously expensive, and often they're done unnecessarily in terms of the expense and the time phase incurred, the time delay incurred in creating a successful trial. So my instinct is that they can be accelerated in terms of reducing the complexity of the trials and accelerating the pace with which one gets definitive answers about the clinical efficacy of compounds under treatment.
Thank you. Another one here.
Good morning. Thank you very much for this very inspiring sharing. I'm very impressed by seeing that actually the lifestyle, change in lifestyle actually is far more effective than treating a cancer.
Vastly.
Are there other factors that we should look at, for example, food, which I will be talking tomorrow, and mental health as a prevention to cancer development?
Well, I don't know. I think the mental health cancer development is a little bit sketchy at present. but food isn't. If you look at the rates, a famous case study, the rates of cancer development of Japanese who moved from Japan to Hawaii, and they had, Japan has a very different spectrum of cancer susceptibility than the United States. Within one generation, the spectrum of cancers that the now Japanese Americans suffered from immediately approached that of their American, Native American, not Native, American Native population. And this only shows you how profoundly important it is, the environment almost certainly being almost entirely diet. And so it's probably the case that 60 to 70% of cancer, if one looks at all the etiologic factors, the causative factors, is preventable. People don't want to know that. If I talk with my colleagues about epidemiologists as being the most important members of the cancer research community, they smile, they mumble, and they walk away, thinking how wrong-headed I am, but it happens to be the case. The future of cancer, of reductions in cancer mortality, comes from the epidemiologists, largely from diet. For example, eating the healthy American diet actually is a relatively quick road to the grave. And people have to recognize that and recognize that the vegetarians will outlive us all, that it's good to be slim. Listen to what I say, not what I do. And get exercise. But most important, eat a healthy diet, a Mediterranean diet. So again, that's not real basic cancer research, it's prevention. But I emphasize for the 14th time this morning, prevention is ultimately far more efficacious than is treatment in reducing mortality from a disease. So I don't know whether you consider that optimistic or pessimistic.
Thank you, Doctor. If I recall, by the way, tell me if I'm mistaken, in an updated version of your famous paper, Hallmarks of Cancer, new hallmarks were added.
Yes.
And one with inflammation and connection with the lipidomics.
Yes.
Which was very close to nutrition.
Yes, absolutely. In other words, we know that, for example, eating a lot of red meat is not so good for you, but we don't really know why it's not so good for you. And here, the big advances will come in studying the messy biochemistry of cells in the body and the microbiome. What are the bacteria living in our gut do to process the foods that we present them with? I think microbiome research is exploding right now, and it's going to give us ways of improving our lifestyle, because we can begin for the first time to understand mechanistically why it is that doing certain things or eating certain things isn't so good for you in terms of leading a long, healthy, and possibly happy life. I don't guarantee the happiness, and I don't guarantee that treating chronic depression is going to reduce, for example, cancer incidence.
Thank you very much, Robert Weinberg. Thank you very much for having me.
Thank you for having me.